Courtship, mating, reproduction. The cycle begins in the case of the ringdove with the male's unique bowing and cooing, or bow-coo. And much ingenuity has been used to observe this species, which serves as a model for learning about all reproductive behavior. Scientists, using computers and television, watch the rituals of the collared dove's breeding cycle. Studied since the turn of the century, the ringdove's habits are well known. What motivates them is still a challenge to researchers. In the heart of New York City, at Columbia University's Barnard College, some of the most detailed research into the behavior of the ringdove is carried out. A black neck ring characterizes the species Streptopelia rissoria, and both male and female have this color. The ringdove adapts well to the confines of the laboratory, mates and breeds almost continuously. The doves are monogamous. Once they choose a mate, they remain faithful throughout their lifetime of some 15 years. The bow-coo identifies the male. With his proud strut and his call, he courts the female, who, coy at first, avoids him. But her resistance gives way, and she signals her acceptance by preening. For the next few days, the couple preen together and start their search for a nest site. The male's head down, tail up posture indicates that he is nest soliciting. For purposes of identification, the male doves are marked with yellow on their wings and foreheads. After days of courting, it is the female who signals her readiness to mate. She begs for food, and the male gently feeds her from his crop, an act that much resembles kissing. They enter the last stage of the long courtship ritual. The female solicits with a graceful crouch, but the male does not respond. Again she begs for food. Again he feeds her. Again she crouches before him. He accepts, and they mate. They continue to admire and preen each other as they enter the next phase of the cycle, nest building. During his survey of possible homes for his young, the male returns most often to solicit at the nest bowl, which substitutes for a tree branch. He indicates his choice by holding his oblique position while cooing and swiftly vibrating his wings. Once the couple has settled on the nest's location, the male brings straw and twigs to the female. She does the actual nest building, which takes about 3 days. In this nest, the male dove will share with his mate the responsibility for the welfare of the eggs and for the care of the young. The ringdove's clutch consists of 2 eggs, and for the next 15 days, until the eggs are ready to hatch, the male, as well as the female, will take a turn on the nest each day, keeping the eggs warm and moist. A rhythm of sharing characterizes the parental behavior of the ringdove. At the psychology department of Barnard College, Dr. Ray Silver is in charge of an extensive examination of the role hormones play in the ringdove's breeding behavior. The laboratory computer stores the data collected by means of closed-circuit television. On videotape, the courtship, nest building, and incubation of several pairs of birds is recorded without disturbing the dove's environment. Ring doves grow quickly and adapt well to laboratory experimentation. Their development from newborn squab, blind and unable to feed itself, to fully developed, self-sufficient fledgling takes only 21 days. A large colony of ring doves, birds of all ages, is maintained at the psychology department of Barnard for the purpose of behavioral studies. Hormones have long been known to affect reproductive behavior. Dr. Silver's studies involve 3 procedures: to remove the hormone and observe if the behavior disappears or declines, to replace the hormone to see if the behavior reappears, to measure hormone levels when the behavior appears and disappears. When all 3 steps point to the same results, a given hormone can be said to produce a particular effect. It has long been established that ovulation and pregnancy in the human female are times of major hormonal change. Ray Silver is studying hormonal changes during the parallel times in the ring dove's life, ovulation and incubation. To study the relation between female hormones and behavior, the first procedure is to remove the bird's ovaries. By removing the organs which produce the hormones, namely estrogen and progesterone, their flow into the bloodstream of the female dove is stopped. When eggs are provided to a female whose ovaries have been removed, she does not incubate. She fails to take her turn in warming and protecting the eggs. We have seen that removal of the sex hormones does affect the incubation pattern of the female. The next step is to replace the hormones, in this case by injection. When appropriate levels of both estrogen and progesterone have been replaced, the parental behavior of the female is again observed. Once more, the indication is that the female hormones affect the dove's incubation behavior. She now covers the eggs with her body to keep them warm during the pre-hatching period, sharing in her normal way this duty with her mate. To determine if there is normally a correlation between the dove's sex hormones and parental behavior, hormone levels in the blood of both female and male doves are carefully measured and charted. Centrifuging the blood sample separates the red blood cells and the blood plasma, from which the hormones can then be isolated. A known amount of radioisotope is added to the unknown quantity of each hormone. The scintillation counter gives an accurate measurement of each sample's radioactivity. From this count, the hormonal level at each phase of the reproductive cycle can be calculated. The results of the radioimmunoassay of female hormones, when charted, show that the estrogen level of the female dove goes up during courtship and declines during incubation. Progesterone secretion shows a similar pattern, peaking around ovulation. Thus, all 3 kinds of experiment, removal, replacement, and assaying ovarian hormones, indicate that female hormones mediate the transition from courtship to incubation. Radioimmunoassay gives us a secretion pattern of normal male doves during the reproductive cycle as well. While testosterone levels rise during courtship, progesterone levels do not change in any systematic way related to behavior. During a complex series of experiments involving removal and replacement of male hormones, the absence of testosterone decreased the male's inclination to bow coo and charge the female, but did not eliminate his ability to incubate. Males castrated several days before pairing continued to nest solicit and nest build, seeming to take their behavioral cues primarily from the actions of the female rather than from hormonal secretion. With the source of the hormones, the testes, recently removed, the male ringdove continues to take his turn incubating the eggs. A special cage was devised to observe the day-to-day cooperation of the parents during the 2-week incubation period. On closed-circuit TV, the daily sharing of responsibility for the eggs can be viewed and recorded on videotape. Several pairs of incubating doves are monitored simultaneously, and information on their patterns of behavior is stored in a computer. In the early morning, it is always the female sitting on the nest, protecting the eggs. At about 10 o'clock, a nest exchange takes place. The male arrives, often bearing a piece of straw as a gift, and he takes his turn on the nest. A regular sitting pattern is soon observed. The male always takes his turn in mid-morning. The female leaves the nest each day during the midday hours. The time of each nest exchange and the duration of each sitting are coded and entered into the computer. A comparison of the nest exchanges of all the pairs of doves shows a regular nesting pattern. This pattern, repeated each day during the 2-week incubation period, finds the male always protecting the eggs during the middle of the day. In the late afternoon, the female regularly returns to the nest to cover the eggs. This second exchange occurs at about 4 o'clock in the afternoon. After coaxing the male off the nest, the female settles down to keep the eggs warm throughout the night. Data analysis reveals that all of the incubating pairs being observed have similar time-sharing schedules. From the day the eggs are laid, the male always takes his turn in the morning and sits for a block of time in the middle of the day. The female sits for the rest of the day and through the night. Once established, the regular schedule is followed by all of the pairs of doves. Throughout the 15-day incubation period, the nesting pattern was repeated day after day. If during the incubation period, a male is paired with another male, they both try to nest during the midday hours, fighting to take their turn on the nest during that block of time. Paired females do not actually fight, but they do not establish any regular pattern of sitting. To further probe the dove sitting pattern, some ingenuity was needed to turn night into day and day into night. The scientists used constant dim red light to replace the normal cycle of daylight in the dove's environment. The incubating doves no longer can detect the time of day from external cues, but they continue to follow their normal sitting pattern. The experiment reveals that the doves possess an internal biological timer, a genetic clock. A computer readout shows that the male continues to sit for a midday block of time, with a drift or free run the only alteration of behavior from his normal pattern. Another experiment investigates the dove's time-sharing schedule still further. The objective is to interrupt the normal sitting pattern and observe the results. In a special cage, access to the nest is controlled by a sliding door. The closed gate prevents the male from joining the female at the time of a normal nest exchange, and the male paces impatiently, while the female remains on the eggs past her normal leaving time. The male continues to pace before the gate during 2 hours of his regular sitting time. As the door opens, the male hurries to the nest to take up his post. Since he has been detained for 2 hours at the start of his sitting period, the scientists watch to see when he will leave the nest. Although the female sat for an extra 2 hours, she returns to the nest at 4 o'clock in the afternoon. But the male tries to stay on the eggs for his full 6-hour block of time. Observation of many pairs of doves reveals that the dove on the nest always tries to sit for its full duration, while the dove waiting to take its turn always arrives at its appointed time of day. When the routine of one dove is interrupted, as in the case of this male, the usually cooperative doves actually fight as they compete to incubate the eggs. Their nest exchange is delayed by nearly 2 hours, the time the male was kept from going to the nest. To determine whether hormones play a role in establishing this rigid parental behavior, Dr. Silver implants silastic pellets filled with crystalline male hormones in female doves. These pellets, inserted under the skin of the doves, will release male hormones into the female's bloodstream over a prolonged period of time. Similarly, a group of male doves are implanted with female hormones. Doves implanted with the hormones of the opposite sex take on the nesting behavior of the opposite sex. Females implanted with male hormones take over the midday block of nesting time, starting at about 10 a.m., the block observed to be exclusively the male's nesting time of day. The implantation of male hormones in a female seems to be the direct cause of her taking on the male pattern of incubation behavior. Here, as in the female's courtship and incubation cycle, hormones can be said to play a decisive role in determining behavior. Through this series of hormone experiments, our insight into the behavior of one vertebrate, the ringdove, has been increased, and with it, our understanding of the delicate interaction between behavior and hormonal change, our understanding of how male and female of one species reproduce and care for their young. The conclusion of the breeding cycle and the start of a new life. From inside the egg, the ringdove squab pecks its way out of its protective covering. Hatching may take several hours, which time-lapse photography condenses into a few minutes. From one egg, a squab emerges. From the second egg, another baby dove will follow a day later. Once again, male and female doves share in caring for the young squabs, as they did for the eggs from which they emerged. The parents continue to take turns on the nest until the young have feathers enough to regulate their own body temperature. Through experimentation and observation, some of the relationships between hormones and reproductive behavior have been established in the breeding cycle of the male and female collared dove, thus extending our knowledge of mating and parental behavior of all creatures, including man. The young grow quickly, feathers gradually replace down, but the squabs remain dependent upon their parents for food. It will take 3 weeks for the young fledglings to become self-sufficient, and then the parents will resume courtship and the cycle will begin anew.